this video, we are going to cover the physiology behind what hearing is and equilibrium. So first, let's talk about the ear. You know, we have an outer ear, and that outer ear is made up of what we would refer to as the pinna. So this is that thing that, you know, you cup when you want to hear things better, or, you know, the little lobe of the pinna is where you stick your earring at. And then it's also really important to collect sound waves, and that's why you cup your ear, because it helps you to collect those waves better. Or if you've ever watched an animal where they turn their head in one direction or another in order to be able to collect all those sound waves going into their ear. Now, off of the pinna, you have the ear canal, or sometimes this is also referred to as the external auditory canal. And this is where the sound waves are going to travel, and then they're going to hit what we refer to as the tympanic membrane, or the eardrum. Now, the middle ear first consists of the eustachian tube, and that it can also be referred to as the auditory tube or the pharyngotympanic tube. Now, this is the tube that goes from the back of your throat all the way to the middle ear here. So you have the external auditory canal here, and this is that tympanic membrane we mentioned before. And from the tympanic membrane into this area, we refer to this as the middle ear. So you have the outer ear, you have the middle ear, and then we'll talk about later this cochlea area. This is the inner ear. So the eustachian tube, or the pharyngotympanic tube, it starts at the back of your throat, kind of along the back of your jawline, and it goes into this middle ear here. Now, normally this tube is collapsed, okay? But the whole reason that we have this tube is because it can open, and as it opens, it helps to normalize pressure. So, you know, if you are driving up a hill or down a hill and you feel your ears popping, that's that equilibrating or normalizing of the pressure in your middle ear with the pressure outside in the atmosphere. Now, one of the other things I'd like you to know about the middle ear is that there's potential for things to get up through the eustachian tube into that middle ear. Now, usually this doesn't happen on a daily basis. However, think about, let's say you have an individual who, I don't know, let's say they have a really bad abscess tooth, and they haven't had a chance to get to the dentist yet. And now they're sleeping, and this infection from the abscess tooth is draining towards the back of their throat. Now, as it drains toward the back of their throat, some of that infection can actually get up through this pharyngotympanic tube and into the middle ear. And that can create eventually what's referred to as otitis media, and otitis media just simply means middle ear infection. Now, notice also right here there is this opening. This is called the mastoid antrum. You can see this here. And right here, this is your mastoid process. So if you feel behind your ear, kind of behind your earlobe, and you feel that little bump, okay, in the bone, that's your mastoid process. And that mastoid antrum opens up into that bony area, that mastoid process. Now, the mastoid process is what we call a sinus in the bone, and that basically means that area of the bone is hollow. So now, you have a person who has an infection in their tooth, and that infection is draining into the middle ear, and they can get a middle ear infection or otitis media. If that middle ear infection is bad enough, the infection can then drain into that mastoid antrum, that open area, and the infection can drain into the mastoid process, and now the individual will have what we call mastoiditis. Now, if it's bad enough, you can tell the mastoid process begins to swell up, and I've seen swellings that are about like the size of a softball. And by that time, however, the infection has drained out of that mastoid process and onto the brain. And so, honestly, problems with the teeth and infections in the mouth can, if bad enough and not taken care of soon enough, can actually cause infections in the brain and potentially can even kill someone. Now, the tympanic membrane, or the eardrum, is what we're looking at right here, and you can see this membrane is pointed towards the middle ear. And what happens is, as air is coming through this canal, it's going to hit this eardrum and make this eardrum vibrate. Okay? And we'll talk about how that vibration occurs. When the tympanic membrane vibrates, then you have pushing on these three bones here, which are also referred to as the ossicles. Now, the first of these little tiny bones is called the malleus, or also referred to as the hammer. And then the second bone is the incus, and the hammer hits on what's referred to as the anvil. And then the stapes, or it kind of looks like a horse's stirrup. And the whole job of these bones is to amplify sound. And so we'll talk about the fact that sound is amplified 20 times its original sound as it goes through the ear canal. So again, we have the malleus, and then the incus, and the stapes, and these are the three smallest bones in the human body. So we're going to talk about sound, and so the question is, how does sound work? So when we clap our hands or you strum the string on a guitar, what's happening is air is beginning to vibrate, okay? And as the air vibrates, these vibrations create sound waves. So literally, think about a roller coaster going up and then coming back down again. That's a type of sound wave. And that wave, okay, carries vibrations through the air and to our ears. So the guitar or our voice, those sound different because they produce different types of sound waves. So in this picture, you can see this is what I'm talking about by that wave. So imagine you're going up the roller coaster and then back down again. And different types of sound produce different types of waves. Now when we talk about frequency, this is the number of waves that pass a particular point in 1 second. So if you could see a wave, a sound wave, something traveling in the air, and you could maybe hold up a pencil and you could see those waves, the peak or the top of the wave, every time it passes by that pencil. That would be one sound wave or one wave, okay, passing by the pencil. Now, if you wanted to measure the frequency, it's going to be how fast those waves are passing by your pencil, okay? So if in 1 second you have 5 waves or 5 peaks of waves passing by your pencil, then your frequency is 5. If in 1 second you could count 50,000 peaks of waves passing by your pencil in a second, your frequency is 50,000. So in the top picture here, you can see low frequency, and you can see how these waves would be traveling slower. And at the bottom picture, this is going to be high frequency. These waves are traveling much faster. So frequency has a lot to do with speed of travel. It also has to do with wavelength. So wavelength is the distance between two peaks. So if we're looking at our waves and you have the top of each of those waves or the peaks, how far is it? What's the distance? This is our wavelength. So the longer the wavelength, it gives you a lower frequency. And so you can see in the picture at the top here, you have this much longer wavelength. That simply means that there's a greater distance between these two peaks. Whereas you have a higher wavelength in the picture at the bottom here, and that's because between those two peaks, there's less of a distance. Now, frequency tells us the pitch of a sound. So if we have a low frequency wave, then we have a low pitch sound. If we have a high frequency wave, we have a high pitch sound. So for instance, the picture at the top might be somebody who is singing bass and they have a very deep, low pitch voice, whereas the picture at the bottom, the high frequency wave, you have somebody who's singing soprano, so that they have a very high pitch voice. 1, 2, 3, ready, go. So the lower the frequency of a wave, the lower the pitch of the sound. The higher the frequency of a wave, the higher the pitch of the sound. And when we're measuring pitch or we're measuring frequency, we measure this in what is referred to as hertz. Adults can hear a range of frequencies between 20 to 20,000 hertz, whereas children can hear a greater range between 50 to 50,000 hertz. So the older you get, the lower your range of frequencies is going to be. Now, amplitude. This is how tall a wave is, so the height of the wave. So amplitude is measured from the trough of the wave to the peak of the wave. And the trough is the lowest part of the wave. The peak is going to be the highest part of the wave. So the shorter the amplitude of the wave, the softer the sound is going to be. The taller the amplitude of a wave, the louder the sound is going to be. So loudness is measured in decibels. All right, so let's talk about the inner ear, which is also referred to as the cochlea. And the inner ear is made up of several sections. The first section is referred to as the scala vestibuli. And this is the top canal. So if we were to take that cochlea and totally unwind it so you don't have that snail shape anymore, the top canal here is the scala vestibuli. And this is filled with what's referred to as perilymph. And then you have what's called the scala media, and this is the middle canal here. And it's also referred to as the cochlear duct. And we're going to talk about this in more detail. It contains the neurons for hearing, and it's filled with endolymph. The scala tympani is this bottom canal here, and this is also filled with perilymph, and it is continuous with the top canal or the scala vestibuli. So we're going to talk about each of these, and we'll talk about how sound is traveling to be able to stimulate the auditory nerves for hearing. Now, in this first picture, you can see the external auditory canal that we talked about, and then you have the middle ear, and then again, this inner ear has been unwound so that you don't see that snail shape anymore. Then we also have the tympanic membrane, and that's separating our outer ear from our middle ear. And in the middle ear, we have those three small bones, the malleus, the incus, and the stapes. Okay, so what I've added here is what's referred to as the oval window. And the oval window right here is the area between the inner ear and the middle ear. So you can see right in this area, this is the oval window. And then right here, this is what's referred to as the round window. And so you can see the round window right in this area here. And then the cochlear duct is here in red. And then remember, we have the scala vestibuli, which is in the upper region here. And then you have the scala tympani, which is in the lower region down here. And these are both filled with perilymph. And then inside the cochlear duct, this is filled with endolymph. Now, there's not a lot different with perilymph versus endolymph, except there's a little bit of potassium and a little bit of chloride difference. At the top of the cochlear duct here, you have this really thick layer. And this layer is kind of a jelly-like layer. And this layer, we refer to this as the tectoral membrane. Let me write that out here. So this is the tectoral membrane. One, two, three, ready, go. Then down here, there is another layer, and this layer, it's not so jelly-like, but this layer is what's referred to as the basilar, and basilar basically means bottom. This is the basilar membrane. Okay? Now, embedded in the tectoral membrane are the dendrites of our neurons for sound. So let me take a second and draw these dendrites. And so remember that tectoral membrane is that gooey, sticky type of membrane, and these dendrites are all stuck inside that membrane. Now, I'm not making a whole bunch of these neurons. There are literally thousands of them, but you get the idea. Now, also coming out of the basilar membrane, there are these cells, and these cells are helping to hold the dendrites in place, because remember, this is a really fluid environment here, and these cells are in between each of these dendrites, and there's a few layers of these cells, and these cells are referred to as supporting cells, because that's just what they do. They're supporting cells. All right, so now you have all the basic structures that we need to talk about when it comes to hearing in the ear. So what happens first is you have some type of sound wave that's coming in this external auditory canal. And this wave, of course, is traveling at a certain frequency and a certain amplitude, and it's hitting on the tympanic membrane, that eardrum. And the eardrum is vibrating. Now, as the eardrum vibrates, it's going to start pushing on our first bone here, which is our malleus. Now, the malleus, as it starts to move, it begins to push on the second bone, which is our incus, and then the incus starts to push on the third bone, which is our stapes. Now, the stapes because of the way that the malleus, the incus, and the stapes are set up, when the stapes starts to move, it starts to push on the oval window here, okay? So we've got this oval window, and the stapes is pushing on it. Now remember I told you that the job of the malleus and the incus and the stapes is to amplify the original sound wave by 20 times. All right, so now what this means is, imagine you have somebody in a swimming pool, and you say to this person, okay, I want you to make the biggest waves you possibly can, and if you make the biggest waves you possibly can, I'm going to pay you $1,000. Now, you don't expect that that person is just going to then blow on the water, okay? If they just blow on the water with their own breath, they're not going to be able to make any ginormous waves. So instead what they do is they grab a big inner tube and they get inside that inner tube and they start bouncing up and down inside that swimming pool with that inner tube. And by bouncing up and down, it's able to push on the water and start to create big waves. So this sound wave that's coming into the external auditory canal is like the person just blowing on the water. Whereas the three ossicles, the malleus, the incus, and the stapes, what they are is they're like the inner tube, and they're starting to push on that oval window. And if you remember, behind that oval window is a fluid. It's the perilymph. And so now what's happening is from that oval window, we start to get the fluid moving, and it begins to move in waves. Okay? So this fluid is moving in waves. Now, as the fluid is moving in waves, what it's doing is it's actually causing the tectoral membrane to also move. Because remember, the tectoral membrane is this kind of jelly-like substance. So the waves are passing over top of this tectoral membrane, and it is moving back and forth. And now embedded in this membrane are your dendrites from your auditory neurons for sound. And so kind of think of those dendrites as seaweed. Okay, so you have the ocean that's moving back and forth in waves, and the dendrites are moving with those waves back and forth. So now, as these waves here are being created by the oval window and by the ossicles, they're moving across the top of the tectoral membrane and causing it to move back and forth with the waves, and that's causing the dendrites to also move back and forth. And it's the movement of the dendrites, what we actually call the shearing of the dendrites, that open sodium gates. And sodium starts to diffuse into this neuron, and we start to send neurological information to the brain. Now the big deal is, how does the brain know the difference with the pitch of the sound? Well, remember, pitch has to do with frequency, and the higher the frequency, the higher the pitch. The lower the frequency, the lower the pitch. Now, if we could take this cochlea with all its neurons and divide it into 3 equal parts, okay? So we're going to divide it here. It's going to be part 1. And then we've got part 2, and then we have part 3. If you're looking at a really high frequency or high pitch sound, one of the things we know is that this high frequency, high pitch sound doesn't travel in the perilymph very far. It dies out about 1/3 of the way through. And so that means that the neurons in this first third of the cochlea are the only neurons that are going to be stimulated. And if only the first third of the cochlea, only those neurons are stimulated, the brain perceives this as a high-pitch sound. Whereas when we're talking about a medium pitch, this is going to travel a little bit further, but end up dying off about 2/3 of the way through the cochlea. So now we have neurons in the first third of the cochlea and neurons in the second portion of the cochlea. And when those two portions of neurons are stimulated, the brain knows this is a medium pitch sound. Whereas you saw when I drew it in blue, that wave went much further. That's because it has a very low frequency, and low frequency waves are able to travel much further, stimulating all three portions of the cochlea. So the neurons throughout the entire cochlea are going to be stimulated by a low-pitched sound, which then also means that the brain is going to be able to perceive when all the neurons are stimulated, we've got a low-pitched sound going on. So one other thing I want you to notice is that in this first third right here, these neurons are being stimulated by low, medium, and high frequency waves. And so these neurons are working more than the neurons in the other 2/3 of the cochlea, and these neurons are the ones that are going to wear out faster. So usually as we get older, the sound that wears out the fastest is going to be high-pitched sound. So it's the sound we're most likely not going to be able to hear as well. Now, all of these axons here that are coming off of our neurons are going to form the auditory nerve, because you know a nerve is a bundle of axons. And so this is going to be our auditory nerve. Which is then going to take information to the temporal lobe of the brain so that we can perceive sound. So now you know that when we're talking about loudness, we're talking about amplitude, okay? And when we talk about a high amplitude wave versus a low amplitude wave, it's kind of like thinking about a high amplitude wave in the ocean versus a low amplitude wave in the ocean. So if you're standing in the ocean enjoying yourself and a little 2-foot wave comes and hits you, it's no big deal. It doesn't really move you very much. On the other hand, if you're standing in the ocean and a 20-foot wave comes and hits you, it's going to move you a whole lot. And that's the same thing that happens in the ear. When we have a higher amplitude wave, it causes the tectoral membrane to move a lot more. And as it moves, it pulls on those dendrites more. And the more we get those dendrites to bend, the more sodium gates open. And the more sodium gates open, the more information is sent to the brain, and the brain perceives this as loudness. So you can see here, these are our auditory neurons. This is our tectoral membrane, and the auditory neurons are taking these signals up through the medulla, up through the midbrain, and then into the temporal lobe of the brain so that we can perceive what it is that's sending sound waves into our ear. Now, the next part is equilibrium. And so in the cochlea, we have the ability to perceive static equilibrium and also dynamic equilibrium. So this inner ear has two portions to it. We have the area here, which has the cochlear duct in it, and then we have this area here, which is going to help us to perceive static and dynamic equilibrium. So static equilibrium is where you're keeping your equilibrium, but you're not really doing very much movement. So you're either sitting in a chair or you're standing up straight and you're not really moving, but you're also not falling over, okay? So this uses a region called the vestibular region of the inner ear. And when we look at this vestibular region, we're looking at this portion right here, okay? And so when we look at dynamic equilibrium, we're actually looking at this portion here, okay? And this is where we have greater body movement. So you're running, you're spinning, you're rolling, whatever it is, but you're keeping your equilibrium. And we're using what's called the semicircular canals. Now with static equilibrium, okay, we're using that vestibular apparatus, and because we're not really moving a whole bunch, we also refer to this as linear acceleration. So you can see here, this is the vestibular region. You have a region called the utricle and the saccule, and they are composed of something kind of similar to what we saw in the ear. So we have our neurons, okay, and these are our dendrites attached to the neurons. These are our supporting cells, and then we have this membrane on top. And this membrane, very similar to the tectoral membrane, is a gel-like substance we call the otolithic membrane. And on top of the otolithic membrane are these calcium carbonate crystals, and these are referred to as otoliths. And this is all referred to as the vestibular apparatus. Now, how this works is, as you're doing a little bit of movement, okay, the calcium carbonate crystals of these otoliths are going to be pulled by gravity. So if you move your head, let's say to the right, or you move your head to the left, or you move your head forward or backward, but you're not really moving your body a whole lot, these crystals are going to be pulled by gravity. And when they're pulled, they're going to pull on that gel-like otolithic membrane. That membrane is then going to cause these dendrites to bend. And depending on which direction the dendrites bend, it will send information to the brain telling the brain which way the body is moving. And then the brain can compensate for this. Now with static equilibrium, we're using those semicircular canals, and they're sensing what we call rotational acceleration, but that means the body's moving a whole bunch, and we're going to have to do something to keep the body from falling over. So inside these semicircular canals, you have endolymph, so it's filled with a fluid. Now, if you see here, the canals are situated in 3 different planes, which means that any way the body turns, one of these semicircular canals are going to be able to pick up that turning motion. And at the base of these canals, you have this region called the crista ampullaris. And the crista ampullaris is made of a gel-like substance here. This is the cupula. And embedded in the cupula are the dendrites, okay, for the vestibular nerve, which is going to help us maintain our equilibrium. So this endolymph is going to be able to move as the body moves, and it's going to circulate around these canals. So the endolymph is going to hit on this cupula, this gel-like substance, and cause the gel to bend in a certain direction, bending the dendrites. So depending on which canal is having the endolymph move and which way this cupula is moving, it tells our body this is the direction the body is moving in. You need to do what's ever necessary to maintain equilibrium. All right, thank you for listening to the Hearing and Equilibrium lecture.